Good morning, we are going to talk about enhanced LRP8 expression induced by Helicobacter pylori drives gastric cancer progression by facilitating beta-catenin nuclear translocation.
Helicobacter pylori is a bacterium that affects the mucosa of the human gastric epithelium and has the ability to live in an acidic environment. It affects 2/3 of the world population, and its infection has been associated with gastric cancer. Gastric cancer is the form of common neoplasm worldwide, and it is caused by an abnormal growth of cells in the inner layer of the stomach. Nuclear beta-catenin is a protein involved in cell adhesion and nuclear signal lines, and it is relevant to certain types of cancer by maintaining the structural integrity of many epithelial tissues.
This diagram shows how Helicobacter pylori promotes gastric cancer through a molecular pathway. Helicobacter pylori releases CAGA and beta-ADP epitopes, which activate LRP8, causing beta-catenin to move into the nucleus and trigger genes that promote cell growth, cancer cell stem, and chemotherapy resistance.
So, the overall objective of this study is to investigate the potential role of LRP8 in Helicobacter pylori infection and gastric carcinogenesis.
Methods: immunohistochemistry. This antibody-based technique detects and measures proteins in tissues, revealing their location and abundance. It showed that LRP8 is overexpressed in gastric cancer, particularly when tissues are infected with Helicobacter pylori. Real-time fluorescence quantitative PCR. RT-qPCR measures gene expression by quantifying messenger RNA. In the study, it showed high LRP8 expression in gastric cancer cells, especially with Helicobacter pylori infection.
ELISA. It is a method for quantifying an immobilized antigen on a solid surface. The principle of this test is that a specific antibody attached to a target antigen. This method was used to measure the concentration of interleukin-8 in the presence of Helicobacter pylori. Immunoblotting. It is a test that allows the detection and characterization of proteins by electrophoresis separation of a mixture of these. This test was used to evaluate the presence of LRP8 in cell lysates and to see the interaction that this protein had with beta-catenin and the virulent factor CAG8.
This graphic is immunofluorescence in gastric organoids. Human gastric organoids were compared non-infected and infected with Helicobacter pylori. In infected samples, increased expression of LRP8 in color green and POU5F1 in color red. A stem cell marker was observed. The merged signal in color yellow indicates that LRP8 is expressed in stem-like cells activated by infection. This graphic is Western blot of LRP8 after Helicobacter pylori infection. LRP8 expression was analyzed in two gastric cancer cells, SGC7901 and MGC803. After infection with Helicobacter pylori for 12 and 18 hours, in both cells lines, the LRP8 band intensifies over time, indicating increased expression. Beta-actin remains constant as a loading control. And this graphic is a staining of human gastric tissues. Gastric tissue samples were compared across three stages: normal, CAG or chronic atrophic gastritis, and DYS is gastritis dysplasia. 1. H&A shows progressive structural change for normal to dysplasia. 2. AHC for Helicobacter pylori absence in normal tissues, present in chronic atrophic gastritis and more intense in gastric dysplasia. And number 3, IHC for LRP8, low or absent in normal, moderate in chronic atrophic gastritis, and highly expressed in gastritis dysplasia. In this graphic, LRP8 protein is notably reduced in CA1 and CA2, indicating that the silence was effective. Beta-actin is used as a loading control to ensure that the total protein amount is comparable across samples. In graphic 2, LRP8 protein is greatly increased in the OB group. Beta-actin used as a loading control shows similar levels across conditions, and this graphic is, in all cases, LRP8, POU5F1, CD44 expression significantly increases when LRP is overexpressed. Statistical significance indicators is P less 0.001 and P less 0.01.
In figure 4a, by silencing the expression of the protein LRP8, there were no changes in level of beta-catenin, but there was a significant decrease of proteins such as cyclin D1 and c-MYC. In contrast, in figure 4b, it is observed that the overexpression of LRP8 results in an increase of these proteins, cyclin D1 and c-MYC, important for cell proliferation. In figure 5H, we can see that silencing LRP8 reduces the expression of cyclin D1 and c-MYC, while its overexpression increases the activity of this protein, contributing to the proliferation of cancer cells. In addition, the virulent factor Cat-A is associated with LRP8 and boosts the activity of beta-catenin in nucleus.
Murata Camilla says CAGA activates beta-catenin by disrupting E-cadherin. Fan Kuch said ADPF-TOS triggers host inflammation, and Shanks confirms ADPF-TOS induced inflammations. All of them support and strengthen the argument of the article.
Molecular biology makes it possible to understand the events and interactions that occur at the molecular level in the organism, which is important for a better understanding of complex diseases such as cancer. In addition, molecular biology techniques allow a more accurate diagnosis by observing changes in DNA, RNA, or proteins. Thanks to this, research has led to the development of various treatments that, by intervening in cellular processes, can delay the development of a disease.